Why, hello there, everyone. It's March 19th. This is information on the coronavirus genome, the SARS-CoV-2, the sequence of nucleotides, the like A, C, T's, and G's of everything. The genes, the protein, assessing how we can lance it, get rid of it, destroy it, burn it, whatever we're going to do to this virus to stop it. There's been some advancements, or at least I heard about them today with the colloquine or something. Looks like there are some promising treatments already to deal with some of the damage that it does to your lungs, but these are repurposing other pharmaceuticals that we have had for different viruses, not necessarily this one exactly. So going over this one exactly is worth it to see exactly how it works. And it's kind of cool. We've broken it down by genes, and it's really like 10 things to learn about. And so I'm going to talk about that today and look at the information in a certain kind of way. First of all, I wanted to make it so that I can add different strains of the virus wherever it's found to the database quickly, starting to set up a gene database and uh, the locus or loci or whatever they say for that. There's a database number for the a session or the whatever the actual string of somebody getting the information of the genome. And there are nice links with NCBI at the National Institutes of Health.gov website. So countries and places going to be adding all of them and comparing them. And just for the purpose of this video, I have Italy and Wuhan from like one from the Wuhan's from December and the Italy's from January. And it's nice to see that it lines up exactly with the Wuhan version having a little more at the end on what's called the 3' end. If you look at just the 3' end, you've got more nucleotides in the Wuhan version. Maybe the virus is shrinking because the Wuhan version's older, or they just did a more extensive job measuring the end of the 3' end, which you could see down here. If you go all the way down, you'll see that there are more A's here. And it looks like there's one more G and C, which is why that's the, if I said if the virus is shrinking as it grows, that would be awesome. I don't do not take that as a scientific assessment. It's just comparing two of these things here. Anyway, so what I want to talk about is looking at all of the different ways to look at this information. Also, what we need to study up on for tomorrow. I realized that I need to take a crash course in understanding what proteins are and how they are made out of the bases of the RNA as they're sent to the cell. There are 10 of them that really we need to go over because of these 12 things here. Let's just look at Italy, for example. Um, let's look at Italy and also instead of looking at the molar mass, I'm going to do something cool with the calculation of the molar mass and say like the mass or the amount of weight of this stuff. We're going to make it a percentage of the total that we're looking at in this given table at this given time. So what that does is that allows us to see, for example, that the majority of this virus is ORF1AB protein. It's 71% of this thing is this ORF1AB protein. Well, do I have a description on that? I'm trying to grab some different descriptions. We can even sort this by description instead of by order to show you kind of what I'm talking about. Let's get rid of gene for a second and go to description of things and get rid of order and bring back gene. And what that'll do is it will show you kind of how we're starting to group this thing and talk about what is in the genome and why we care. So boom, for example, looks like the polyprotein ribosomal slippage thing, which happens to be that ORF1AB is the 71% of this virus that we're looking at. The top and the bottom take up very little. That's the 5' and the 3' end, right? There's something called an envelope protein here, which also doesn't have very many base pairs of RNA. It's called E. There's a membrane glycoprotein called M, which is a little bigger. There's a nucleocapsid phosphoprotein, which is even bigger, this N here. And then I got all these unspecified proteins, ORF10, ORF3A, ORF6, ORF7A, ORF8. These are probably just squiggled up protein lines of DNA that form certain shapes. They're probably a composition or a collection of these together are probably what creates the key that goes into the ACE2 protein in our cells that bonds to this. Something that's about this combination maybe makes it a perfect fit, which is why it fools the cell into allowing it to seep into the cell. Who knows? I'd like to learn about these proteins. So these are the unspecified ones. And then there's a surface glycoprotein. Sounds glyco usually means sugar, maybe. I don't know. I'm going to go back on my Latin and my medical. We're going to learn. Um, and that S, and that's pretty big also. That's 12% of this thing. So different ways to attack things and also understanding what's going on in these unspecified proteins that I'd like to learn about, right? Well, what's cool is as this really starts to grow and I understand what these things are, let's just dig around on something small. ORF6 is a super small unspecified protein. So let's open up ORF6 and just see if anything looks cool. And the answer is, in the order I have it, it's probably not going to look very cool because I changed my colors around. But um, it's going to give you base pairs, and it's going to allow you to see like the percentage of C's and G's and everything. Like, let's move that around, move the strain up to like there. You can see that this thing is, uh, yeah, predominantly A's and T's which bond together, not so many C's and G's. And then you could look at different orders of this 162 and open it up, see where do the A's appear. You could reorder this. Just all kinds of different things to dig up and see what kind of shape this protein makes or how it can be attacked or destroyed if we need to destroy it or whatever. So like where this is coming along because we can now really compare different strains to see if they're exactly the same. We can then start to really learn, for example, what should be on um, what should be on the mind of working for tomorrow here is going to be understanding what ORF1AB is. Like if this virus, the coronavirus, the SARS coronavirus here, happens to be 7 minutes. I don't know, I'm going kind of long, but whatever. If this thing's going to, we got to destroy it in its form, especially when it's in our body. If 71% of it is this one polyprotein, whatever that has ribosomal slippage or something, or that's the main part of it that's like getting copied in our cell, then figuring out how this sequence really plays out and um, where what the order is and how to do something with this protein is probably really just important when it comes to battling the virus. So it's just interesting to open up and see what the composition of it is because it looks like the strains are pretty much exactly the same from different areas. I'll start to add different areas of the different countries and add it to this and see if we find any difference in the order of things, because when you pull the descriptions out of there, when you're just looking at the order, um, there's certainly an order to how it's reproducing and the fact that right after the 5' UTR, the very beginning essentially comes this huge ORF1. It must be like of the things that get copied in our cell, it looks like it starts with this because I believe the way that the ribosomes or whatever it is in your cell that's copying this RNA that gets plopped into the cell, it starts from the beginning, the 5' end, and then goes down. Sometimes, like HIV, I think is the backwards way, and they have to transcribe it upside down or something. This one, I believe, starts at the top. So the first thing it's going to hit is this. So we should not only discuss this because it's the largest part of this virus, but also because it's the beginning of what it's copying. Start the book at the beginning. So looking through this will be and reading up on what this protein is homework for today, and we'll talk about it tomorrow. Until then, let's come up with a cure. I think we can come up with treatment. It looks like treatment is pretty much almost already there. We just haven't fully tested it and know how effective and how side effecty it really will be. Um, but the chloroquine and antibiotic mixture. So the point is we're getting a handle on it, which I think is going to change the outlook on things. But until then, we all got to stick around and hunker down, I believe, because you don't want to get it and get coughed on, get it in your lungs, and develop lung disease. Right now, we still don't know what's going on, and everyone still needs to be on our toes. So we'll continue on and do another video tomorrow as we try to catch up to everybody else who's probably already created a cure for this thing and see how they got there and maybe make it better. So